Hello everyone, I'm Kaoruko Kikuchi. I'm from Japan and Azabu University. I'd like to talk about my research, reprogramming of canine hepatocytes to hepatic progenitors using small molecule compounds. I have no financial relationships to disclose. First of all, I will talk about liver diseases and their treatment. With the extended lifespans of canine, the number of cases of cirrhosis and severe hepatitis is increasing. The treatment of liver diseases is mainly symptomatic in veterinary medicine in Japan. Although the transplantation is a radical treatment for severe liver failure, it's not performed in Japan due to ethical issues for donor animals. Second, I will discuss some issues in drug development. Second, I will discuss some issues in drug development. About 95% of drugs are metabolized in liver. Drug-induced liver injury is a major cause of drug discontinuation and market withdrawal. The issue in vitro, primary cultured hepatocytes can be cultured long-term and maintain function. Strain cells have inconsistency in function from lots to lots. And the issue in vivo, more than 4,000 healthy dogs are killed in a year in Japan. To solve these problems, we need to establish a hepatocyte for in vitro toxicity assessment and construct an evaluation system that could serve as an alternative to experimental animals. In other words, we need to establish cultured hepatocytes with proliferative potential and sufficient liver function. This is a unique point of our research. We focused on liver progenitors. Liver progenitors can proliferate in vitro and in vivo and differentiate into hepatocytes and bile duct epithelial cells. Recently, it has reported that medium with small molecule compounds enable induction to liver progenitors and maintain function. It means reprogramming is happening in vivo, and research is underway to reprogram hepatocytes to hepatic progenitors in vivo using small molecule compounds and without gene transfection. Let me now present the previous studies in mice and rats. Reprogramming of hepatocyte liver progenitors can be achieved by using 3 small molecule compounds: LOX inhibitor, TGF-beta inhibitor, and GSK3 inhibitors. We call the cells CRIPs by their initials for chemically induced liver progenitors. CRIPs can remature into hepatocytes and bile duct epithelial cells. Furthermore, when transplantation into mice with chronic hepatitis, CRIP cells were highly replacement into the liver, 75 to 90% without tumorigenesis. I will begin with a brief description of the methods used to create CRIPs. A small amount of CRIPs of canine hepatocytes were collected, and the cells were cultured in hepatocyte growth mediums containing 3 small molecule compounds. Assessment of CRIPs were conducted involving morphological observations, quantitative real-time PCR, immunocyte chemistry, and flow cytometry. This slide shows the proliferation of clips. In clips induction groups containing yak cocktails, small hepatocytes appeared and continued to proliferate while forming colonies. On the other hand, in the control groups without yak cocktails, hepatocytes gradually expanded and disappeared. Next, this slide shows a graph of real-time PCR comparing the amount of gene expression of liver biopsy and clips on day 28, alpha-hetoprotein, Sribox9, cadherin-1, keratin-19, which are highly expressed in liver progenitors, are more expressed in clips. On the other hand, albumin, which is a marker of liver function, CYPs, which evaluates metabolism, MRP2, which is a marker of bile duct epithelial cells, and FOXA2, which is highly expressed in mature hepatocytes, or more expressed immediately in liver biopsy. And this is a heat map that briefly shows these graphs. In clips, you can see that liver progenitors markers gradually become highly expressed. This slide shows the result of immunocytes chemistry of clips on day 28. Corresponding protein expression of albumin, MRP2, SOX9 were confirmed. All proteins shows positive results. These are the results of cell surface antigen analysis of flow cytometry. Among the 5 markers of undifferentiated hepatocytes, CD29, CD44, and DLK1 is positive. These results also suggest that the clips were transformed from mature hepatocytes to undifferentiated hepatocytes. Next, I will discuss how to remature clips. As you know, hepatocytes are difficult to maintain function while in culture. However, it's known that 3D culture of hepatocytes increase liver function than in 2D cultures. For these reasons, we performed 2 types of rematuration, 2D culture and 3D culture, and compared them. In addition, hepatocyte growth factors are added to the hepatocyte induction mediums with small molecule compounds. In 2D cultures, for 2 weeks, small clips were transformed into large mature hepatocyte-like cells, but the spheroid of clips were incubated until about day 10, which are change of culture plates on day 4. The clips gradually aggregated and transformed into spheroids on about day 3. This result was a little later than I expected. This is the result of gene expression in 2D and 3D cultures of rematured clips. The yellow and orange graphs are associated with the remature of clips on day 16, and the light blue and blue graphs are associated with the cells of day 28. Hepatocyte markers sorted by red frames, such as albumin and CYPs, are highly in 3D cultures than in 2D cultures. In both results of day 16 and day 28. And this graph compared the previous result with the gene expression levels of normal liver and long-term clips. Surprisingly, these results show the 3D culture clips are closer to the gene expression of normal liver. We have successfully induced differentiation of hepatocytes into liver progenitor-like cells. Clips can proliferate, and an increase in liver function was observed by 3D culture. We plan to further investigate culture conductions and conduct pharmacokinetics and toxicity studies on CRIPS in the future. We hope that through CRIPS, we aim for a future where liver regenerative medicine can be safely offered as one of the treatment options, and a future where even one more life is not lost. I'm deeply grateful to be able to participate in such a wonderful conference on my birthday. Thank you for your attention.
Did you say that it's your birthday?
Yeah.
All right, that's great. Congratulations on a great talk and a great day. So how long can you keep those clips in culture, and how stable are they over time? How long can you keep the clips in culture? How many passages?
I try to culture the clips for long term until 42 days, but the clips are little decrease the function. So I think the most suitable for maintaining clips function is day 16. Okay.
More questions?